In Unit 3, Lecture 2, Part 2, we're going to be overviewing parts of innate immunity, namely phagocytosis, the complement pathways, and antibacterial peptides. Now, the next two SoftChalk lessons we covered deal with the ability to resist innate immune defenses and the role that it plays in helping bacteria to colonize the body. But before we can understand how bacteria can resist our innate immune defenses in order to better colonize, we have to understand how these innate immune defenses like phagocytosis, the complement pathways, and antibacterial peptides normally function to protect the body. So in this SoftChalk lesson, we'll be looking at an overview of phagocytosis, the complement pathways, and antibacterial peptides. Once again, don't forget to go over your fundamental statements or bullet points for this SoftChalk lesson and to learn your detailed learning objectives so you'll be ready for Unit 3 exam. Now remember, we're currently talking about virulence factors that promote bacterial colonization of the host, things that microorganisms do, and more specifically in this case, bacteria do, to establish themselves in or on our body. And we've already discussed the first four of these in previous lessons. The next two SoftChalk lessons will look at how the ability to resist innate immune defenses plays a role in helping bacteria colonize the body. Again, before we can understand how bacteria resist innate immunity, we have to understand how innate immunity normally works to protect the body. Specifically, we're going to look at how phagocytosis, the complement pathways, and antibacterial peptides help to protect the body during our innate immune defenses. And we'll start out with an overview of phagocytosis. Now, we did cover this back in Unit 1 when we talked about capsules, but it bears repeating. And eventually, Unit 5, we'll be looking at innate immunity in detail. So as we've learned, the first step in phagocytosis is attachment. That is, the surface of the microbe must be attached to the cytoplasmic membrane of the phagocyte. And it has to be attached before it can be pulled in, engulfed, and eventually destroyed. So attachment of microorganisms is necessary for ingestion, and the attachment can either be unenhanced or enhanced. Now, unenhanced attachment is where pathogen-associated molecular patterns, or PAMPs, things like peptidoglycan, teichoic acids, lipopolysaccharide, mannans, and such that we learned about previously, bind to endocytic pattern recognition receptors on the surface of phagocytes. And we can see that in the illustration in Figure 1, where the surface, remember, of bacteria have these common PAMPs like peptidoglycan, teichoic acids and gram-positives, LPS and gram-negatives, etc. And for all the common PAMPs that our microbes produce, our phagocytes produced endocytic pattern recognition receptors that fit those PAMPs. Now remember, endocytosis is what we collectively call phagocytosis and pinocytosis. When something is engulfed and placed in a vesicle by a phagocyte, we call that endocytosis, taking it in the cell and placing it in a vesicle. And we can see the unenhanced attachment nicely in this animation. So again, all of our microorganisms have some surface PAMPs on them, molecules that groups of organisms share that our innate immune system recognizes. And our phagocytes have endocytic pattern recognition receptors that fit all these common PAMPs. Now remember, when we talked about PAMPs back in Unit 1, we saw that PAMPs bound to PRRs, causing the production of inflammatory cytokines for inflammation. In this case, the PAMPs are binding to a different type of PRR called an endocytic pattern recognition receptor found on phagocytes, and that reaction is what promotes unenhanced attachment, the first step in phagocytosis during innate immunity. So as we see here, the surface of the microbe has some surface PAMPs. Maybe that would be peptidoglycan or teichoic acids on a gram-positive. And our phagocyte here has endocytic pattern recognition receptors that fit that specific PAMP. And that attaches the microbe to the surface of the phagocyte, so it can now be engulfed and eventually destroyed. So since that is just naturally PAMPs binding to endocytic PRRs, we call that unenhanced attachment, a normal part of innate immunity. On the other hand, enhanced attachment is where molecules like the antibody molecule IgG or immunoglobulin G, or some proteins produced during the complement pathways like C3b and C4b, will stick organisms to phagocytes. So molecules like immunoglobulin G, C3b, C4b, and others that we'll learn about later on in the course, molecules that promote enhanced attachment, are also called opsonins. And for that reason, the process of enhanced attachment is also known as opsonization. We talked about that quite a bit in Unit 1. And the enhanced attachment or opsonization is more specific and more efficient than unenhanced. So in Figure 2, we see both the antibody molecule IgG, shown in green, and the complement protein C3b sticking microbes to phagocytes for enhanced attachment. So let's take a look at an enhanced attachment with IgG. So again, during adaptive immunity, our body will make antibody molecules, in this case immunoglobulin G, and the tips of the antibody, the amino acid sequences there, create a shape specific to fit a specific epitope of an antigen on the surface of the bacterium. So the FAB part of the antibody stands for antigen binding fragment. That's the part that actually fits epitopes of an antigen on a bacterium. So here we see the FAB portion of the antibody IgG binding to its corresponding epitopes on the surface of the bacterium, like pieces of a puzzle. And then the FC portion of the antibody then binds to FC receptors for IgG on the surface of phagocytes, so that the antibody is acting kind of like a superglue to specifically stick that microorganism to the surface of the phagocyte. And again, complement proteins like C3b and C4b, shown in Figure 2, can also do that, where the C3b binds to proteins and polysaccharides on the surface of the bacterium, and then the other end of the C3b binds to receptors for C3b on the surface of the phagocyte for enhanced attachment or opsonization. Now, once the organism has been attached to the surface of the phagocyte, the next step is ingestion. And this is done through polymerization and depolymerization of actin filaments, part of the cytoskeleton of our cells. And as the actin polymerizes, that pushes the inner surface of the cytoplasmic membrane of the phagocyte up to produce pseudopods that engulf the microbe, pinch off, and place the organism in a membrane-bound vesicle called a phagosome. So the phagosome is the sacral membrane that forms during phagocytosis, the one that engulfs the organism. And we see that happening in the next animation. So here we see a bacterium that has been attached to the surface of the phagocyte through opsonization with the antibody IgG. And then as actin polymerizes inside the cell, that's going to push the membrane up, producing pseudopods that are going to engulf the organism and place it in a phagosome, as we see here. So there's the organism engulfed and placed in a phagosome. And here's a couple movies showing you time-lapse photography of phagocytosis, in this case neutrophils engulfing some methicillin-resistant Staph aureus. So we'll load that in. So here we see the neutrophils sending out pseudopods that are going to surround this whole mass of Staphylococcus here. And that eventually closes up, pinches off, and places the engulfed bacteria in a phagosome, where they'll eventually be destroyed by lysosomes. Here we see the one down here engulfing some more Staph aureus. And here's another time-lapse photography of phagocytosis. So again, there's a pseudopods of the neutrophil going out, surrounding the Staphylococcus aureus here. And as the actin polymerizes, that pushes the membrane around the bacteria, pinches off, and places them in a membrane-bound sac called a phagosome, and then lysosomes are eventually going to fuse with that and lead to the destruction of the organism. So that's the ingestion, where the organism is engulfed and placed in a phagosome. Now, during the process of phagosome formation, another important thing happens. An electron pump brings hydrogen ions, or protons, into the phagosome. And remember, hydrogen ions are what make acid. So as the protons are pumped into the phagosome, this lowers the pH within the phagosome, so that when the lysosome containing the killing chemicals fuses, the pH is correct for the acid hydrolases, the enzymes the phagocyte uses to break down the cellular proteins of the bacteria to be most active. So these acid hydrolases found in the lysosomes here are designed to only function well at an acid pH, and that protects the cell from its own lysosomes, because the interior of the cell is normally neutral, and the acid hydrolases in the lysosomes aren't very functional at a neutral pH. So by making the phagosome acidic, when the lysosome fuses, that'll be the correct pH for the acid hydrolases to effectively break down the cellular proteins of the bacterium. So here we're seeing a bacterium being attached to the surface of the phagocyte by enhanced attachment or opsonization using IgG. So there's the attachment. Polymerization of actin sends out pseudopods. They surround the bacteria, pinch off, and place it in a membrane-bound sac called a phagosome. Now, right now, the pH of that phagosome is neutral, but it needs to be acidic for the acid hydrolases in the lysosome to effectively break down the cellular proteins. So a proton pump is going to pump in these hydrogen ions, or protons, as we see here, making the contents of the phagosome acidic. So now, in the next step, when the lysosome is eventually going to fuse with the phagosome, it'll be the correct pH. And that brings us to the next step, the actual destruction or killing of the organism. And when it occurs inside the phagocyte, we call that intracellular destruction. So lysosomes, membrane-bound sacs produced by the Golgi complex that contain digestive enzymes and free oxygen radicals and other things that kill microorganisms, is going to fuse with the phagosome, the sac containing the bacterium, producing a phagolysosome, which will then result in destruction of the engulfed organism. So again, the lysosomes have the acid hydrolases and the other killing chemicals like free oxygen radicals. When the lysosome containing the killing chemicals fuses with the phagosome containing the ingested microbe, that forms a phagolysosome, and as we see, the organism is killed and degraded. So here we see the destruction phase in this animation. Here we have a phagosome that's already formed. The pH has already been made acidic inside. So now the lysosomes actually are pulled along the cytoskeleton by little motor molecules that ratchet the lysosome along the cytoskeleton until it fuses with the phagosome, forming a phagolysosome. And now, because of the correct pH and such, the organism is killed and degraded by the acid hydrolases and the toxic oxygen molecules and other killing materials found within the lysosome. So that's intracellular destruction within the phagocyte. But keep in mind that a lot of killing is extracellular destruction by phagocytes. If the infection site has a very large number of microorganisms, and when high levels of inflammatory cytokines and chemokines are being produced, that triggers the phagocyte to basically dump the contents of the lysosomes out of the phagocyte, a process called degranulation, and that kills the microorganisms extracellularly. So this is kind of a shotgun attempt the phagocytes use when there are large amounts of bacteria present. There's too many to engulf nice and neatly. So the phagocytes, as they accumulate, just dump all their killing chemicals and their acid hydrolases and their toxic oxygen radicals and kill everything in the vicinity, including human cells. And that is a body defense. The body is willing to sacrifice some tissue to get rid of a massive infection. So keep in mind as we go through the course, a lot of the damage we actually see as a result of infection is due to extracellular killing by neutrophils, where you have a serious infection with lots of cytokine production. And so the body's defense there then is to just kill everything in the vicinity and try to restore homeostasis later on. And these two animations, if you want to review them again, summarize the whole process of phagocytosis through unenhanced attachment and through enhanced attachment, putting all the steps together. Now, another thing we need to review is an overview of the body's complement pathways. The complement pathways are another key part of our immune defenses, especially in innate immunity. And again, we're going to take them up in detail when we get to innate immunity in Unit 5. But we need to understand at least the benefits of the complement pathways in body defense. And in fact, we learned these briefly also back in Unit 1. We had an objective on stating the 4 benefits of the complement pathway, which we have also in this unit. So one of the benefits of the complement pathway is inflammation. Now, it should be pointed out we have 3 complement pathways: the classical complement pathway, the alternative complement pathway, and the lectin pathway. And we'll take up all 3 of these later on in Unit 5. They differ in how they're activated, but once activated, they all produce the same beneficial complement proteins. And that's what we're looking at here, the beneficial complement proteins from the complement pathway and what they do in body defense, their role. So one of the important things that they do, these complement proteins, is to promote more inflammation. And as we've said quite a few times now, inflammation is how our body defense cells and defense chemicals are able to leave the blood and enter the tissue at an injured or infected site. So a lot of these defense cells like phagocytes and defense chemicals like complement proteins circulate in the blood. But then if you have an infection in the tissue, those defense cells and those defense chemicals have to get out of the blood into the tissue where the infection is, and that's done through inflammation. And 3 complement proteins, C5a primarily, then to a lesser extent C3a and C4a, all lead to vasodilation, where the endothelial cells constrict, the capillaries get larger in diameter, that increases capillary permeability. And that also begins the expression of adhesion molecules on the surface of the leukocytes, or white blood cells, as well as adhesion molecules on the surface of the vascular endothelium. And that enables the leukocytes to adhere to the inner wall of the capillary rather than flowing past the infection site. They then squeeze between the endothelial cells that form the capillary, and they enter the surrounding tissue where they're drawn to the infection site via chemotaxis. So vasodilation, as a result of these complement proteins and other vasodilators, will make the capillaries more permeable so that the defense cells can adhere to the inner wall of the venules, which is where this actually occurs for body defense. And also the vasodilation enables defense chemicals in the plasma or liquid part of the blood to leave the blood and enter the tissue. Defense molecules like the antibodies we make during adaptive immunity, the complement proteins, etc. C5a also causes neutrophils to release their killing chemicals for extracellular killing. So to give you the idea of vasodilation again, this shows you a normal the capillaries of a normal vein, the endothelial cells of a normal venule. And under normal conditions, these endothelial cells that form the wall of a capillary and a venule are butted up against one another. And the plasma and the red blood cells, not shown here, and the white blood cells, of course, just flow through the capillaries and through the venules. So, of course, if there's an infection down here somewhere, outside of that venule, we have to get these defense cells and these defense chemicals out of the venule into the tissue at the site of infection. And that's what vasodilation does, as we see in the next animation. So as the endothelial cells constrict, as we see here, due to many factors, including the complement proteins we just mentioned, C5a, C3a, C4a. As the endothelial cells constrict, that increases the space between the cells, and that makes the venules more permeable. So now the plasma, which is the yellow liquid in here, the yellow, will be able to flow out from the venule into the surrounding tissue, delivering defense chemicals, and then the white blood cells will begin to roll along the inner surface of the venule, eventually adhere, and then squeeze out of the venule between the endothelial cells and crawl to the site of infection. So here we see vasodilation occurring as a result of the endothelial cell constriction, making the venule wall more permeable. And now we see the white blood cells like neutrophils rolling along the inner wall, adhering, and then squeezing out and going to the site of infection. So that's how we deliver the defense cells where they're needed, through vasodilation via the inflammatory response. And the yellow we see leaking out is the plasma, the liquid part of the blood, now called tissue fluid once it leaves, and that contains defense chemicals like complement proteins, antibacterial peptides, and of course antibody molecules. So one of the benefits of the complement pathway, especially with C5a, C3a, C4a, is vasodilation and increased permeability of capillaries and venules to deliver defense cells and chemicals to the infected area. A second benefit of the complement pathway is chemotaxis of phagocytes. C5a and C3a are also chemoattractants for leukocytes. And so the phagocytes will move towards increasing concentration of chemoattractants, which are the complement proteins being generated at the infected site. And that's what allows the phagocytes then to crawl to the area of infection for the purpose of phagocytosis. There's a couple of animations there of showing more detail of inflammation. This is a nice little movie showing you chemotaxis of neutrophils. That's a neutrophil. Chemoattractants. So there we see them moving towards increasing attractant. So that's a nice little movie showing you chemotaxis, how quickly the neutrophils can reorient themselves and move towards increasing chemoattractant, such as C5a from the complement pathway and other chemoattractants produced during immune responses. There's another movie also showing you that. A third benefit of the complement pathway is that two of the complement proteins are opsonins for enhanced attachment, namely C3b and C4b, as we mentioned. As we said, these are opsonins that bind microbes to phagocytes. So one portion of the C3b or C4b binds to microbial proteins and polysaccharides. The other portion binds to receptors on phagocytes for enhanced attachment. So here we see C3b attached to the microbe, attaching to C3b receptors on phagocytes. So that's a third benefit of The complement pathway is opsonization. And those three benefits we've talked about already, inflammation, chemotaxis of phagocytes, and opsonization, show us that one of the major functions of the complement pathway is to promote better phagocytosis, to get the phagocytic white blood cells out of the blood into the tissue via inflammation, to attract them to the area of infection via chemotaxis, and then to stick microbes to the phagocyte through opsonization. And the fourth benefit is called membrane attack lysis of biological membranes. So a series of complement proteins form what's called the membrane attack complex, or MAC. This puts pores in cellular membranes, resulting in cell lysis. And we do use this to kill gram-negative bacteria that are surrounded by an outer membrane, also to kill virus-infected cells and tumor cells. This won't do any harm to peptidoglycan because it's not a membrane, but it will damage membranes of cells. And so in this last animation here, we see the membrane attack complex forming. So maybe this is a virus-infected cell or a tumor cell, or it could be the outer membrane of a gram-negative bacterium, where we see the bilayer membrane, and a series of complement proteins called C5b, C6, C7, C8, all join in the membrane, and they polymerize C9, which is a pore-forming protein, and as water and sodium enters, that leads to lysis of the cell. So that's called the membrane attack complex, and that can help kill gram-negative bacteria and also get rid of harmful human cells like infected cells and tumor cells. So those are the four benefits of the complement pathway: inflammation to deliver defense cells and chemicals to the area, chemotaxis to attract the phagocytes to the infection site, opsonization where C3b and C4b stick microbes to phagocytes, and then at the end of the complement pathway, it can cause lysis of membrane-bound cells through the membrane attack complex, or MAC. Now, the final part of innate immunity we want to talk about just briefly are antibacterial peptides. The body produces a number of these antibacterial peptides. Remember, peptides are just short chains of amino acids linked by peptide bonds, but these are antibacterial in that they can kill bacteria. That includes human defenses. Defensins are very common antibacterial peptides that can put pores in the cytoplasmic membrane of a variety of microorganisms and damage them that way. Cathelicidins are another antibacterial peptide. So these antibacterial peptides are directly toxic by forming pores in the cytoplasmic membrane of a variety of microorganisms, causing leakage of cellular needs. They can also activate cells for an inflammatory response and promote inflammation. So these types of antibacterial peptides are produced by a variety of cells like white blood cells and leukocytes, by epithelial cells. They also circulate in the plasma and in the mucus to help protect the body. So that's a little bit about innate immunity, specifically the role of phagocytosis, the complement pathway, and antibacterial peptides in innate immunity. So our point here is that one of the ways bacteria can better colonize the body is to resist innate immunity, to resist phagocytosis, or interfere with the complement pathways, or resist antibacterial peptides. So in our next two SoftChalk lessons, we're going to look at virulence factors that enable bacteria to resist phagocytic engulfment, which is what we call attachment and ingestion stages of phagocytosis. And we'll also look at how they can resist antibacterial peptides. And then the SoftChalk lesson after that, we'll look at how they can resist phagocytic destruction once they are engulfed. And of course, don't forget there's a self-quiz at the end you can do to quiz yourself on this SoftChalk lesson.